Now we see what happens when we are in the fasting state. So we are in the fasting state. But notice here, in the fasting state as well, the liver, the brain, and the red blood cells, there is same thing that is happening in the liver and in the red blood cells, whether this is the well-fed state or the starving state. That glucose entering into the brain to make carbon dioxide and ATP, that glucose is making ATP and pyruvate, and then pyruvate is making lactate, and lactate is making pyruvate again through Cori cycle, right? Cori cycle. This is the same in the well-fed state and the fasting state. But now you see the arrow of the glucose is not inside the liver. Rather, glucose is now exported out of the liver. And there is not one hormone that is doing these things. The most important one, glucagon. Glucagon is important for fasting. Cortisol is important for fasting, right? Epinephrine and norepinephrine is important for fasting. Then we have thyroxine, that is important for, T3 and T4, thyroxine and triiodothyronine, T3 and T4, this is T4 and T3. And then another hormone, growth hormone, also is important for the fasting. So you see that there are a lot of hormones that are there in the fasting state. Why is it so? To give glucose to the brain and red blood cells, because brain and red blood cells, whether this is fasting or whether this is well-fed state, follow or gets only glucose for their energy purposes. And if we are not getting glucose, then this is going to create a lot of trouble. So remember, hyperglycemia, no problem, person will survive, but hypoglycemia is going to shut down the brain and red blood cell. So hypoglycemia is more dangerous than hyperglycemia. So now we see how this glucose is exported out of the liver. Right? So formation of glucose from non-carbohydrate sources is called gluconeogenesis. Formation of glucose from non-carbohydrate sources is called gluconeogenesis. We'll study that, but before we study that, we just see that what happens. First of all, that glycogen that was there stored in the liver, that is going to make glucose, and then that glucose is exported out of the liver. But glycogen stores are not going to last for long. They are going to be depleted. So once the glycogen stores are depleted, we need again glucose. But from where we will get glucose? We'll get glucose from alanine. Alanine is amino acid. It's glucogenic amino acids. And from where we are getting alanine? Alanine is coming from all the amino acids, right? So there are actually 18 amino acids that make alanine, right? They are all called glucogenic amino acids. And these amino acids are coming from protein. So protein is broken down in the fasting state, and then that protein makes amino acids, and the amino acids ultimately make alanine, and then alanine enters into the liver, and again it is deaminated to make urea and pyruvate, and then that pyruvate makes glucose, right? So formation of glucose from alanine is gluconeogenesis, and the formation of glucose from non-carbohydrate sources, this is called gluconeogenesis. Similarly, we can get glucose from glycerol phosphate, right? And so the second precursor for glucose synthesis is glycerol phosphate. And from where this glycerol phosphate is coming, you see that this is coming from adipose tissue. This is coming from adipose tissue, right? So fat. This is coming from here. It's making here, and that's coming here, right? And now another thing that can make glucose is lactate. So lactate is making glucose. So there are three things that are making glucose: alanine, glycerol, and lactate. They are also called the gluconeogenic precursors, or the precursors of gluconeogenesis, right? So now we see how liver, muscles, adipose tissue complete their energy requirement in the well-fed state, in the fasting state. Sorry. So first of all, things starts from fat, adipose tissue. Inside the adipose tissue, there is fat. And the fat is broken down by hormone-sensitive lipase. Hormone-sensitive lipase. Right? This is an enzyme which is stimulated by glucagon cortisol, and this hormone-sensitive lipase breaks this fat into fatty acid and glycerol. That glycerol goes to the liver and makes glucose. Now these fatty acids are oxidized. These fatty acids are oxidized to make acetyl-CoA, and then that acetyl-CoA is going to complete the energy requirement, carbon dioxide and ATP. Carbon dioxide in the Krebs cycle and ATP in the electron transport chain. And then this fatty acid enters into the blood. From the blood, it enters to the, fatty acid cannot travel alone, so it needs a binding protein, and that binding protein is albumin, right? So fatty acid goes to the liver. In the liver, it is again oxidized to make acetyl-CoA, and that acetyl-CoA is going to make carbon dioxide and ATP. And another important thing that is going to happen with this acetyl-CoA in the liver is that it is going to make ketone bodies. Ketone bodies are synthesized only in the liver. Note, ketone bodies are only synthesized in the liver, but they are not used by the liver. They are exported out in the blood, and from the blood these ketone bodies go to the muscles. And inside the muscle, they again make acetyl-CoA. And that acetyl-CoA in the Krebs cycle and electron transport chain make carbon dioxide and ATP. So this is the source of energy. Ketone bodies are the source of energy for muscles in the fasting state. Again, these fatty acids are entering into the muscles, and in the muscles, fatty acids are oxidized to make acetyl-CoA, and acetyl-CoA is going to make carbon dioxide and ATP. So energy requirements of the muscles are also met by the fatty acids instead of glucose in the fasting state. And the energy requirement of liver are met by fatty acids instead of glucose in the fasting state. And similarly, the energy requirement of adipose tissue are met by fatty acids in the starving state. Right? You see that this is the big picture. And in that big picture of fasting state, we again see a lot of pathways. And now we see that we studied glycogenolysis. That glycogen is broken down to glucose. We studied gluconeogenesis. Glucose is formed from glycerol, from lactate, from alanine. So gluconeogenesis. We also studied the lipolysis, the breakdown of fat into fatty acid and glycerol. We also studied the fatty acids are oxidized, fatty acid oxidation, which is also called beta oxidation of fatty acids. We will see how. So the fatty acid oxidation gives energy to adipose tissue, liver, and muscles. And we also studied the urea cycle, that is same in the well-fed state, in the starving state, urea cycle. And then we studied the ketone body synthesis. So ketone bodies are synthesized, and then ketone bodies are utilized in the muscles. So this is the big picture. If you don't understand this big picture, it's really difficult to understand the metabolism. If you understand this big picture, how and what is happening in the well-fed state, in the starving state, and how the different organs are getting energy and from which thing. If you understand this, the biochemistry is really very easy thing. And if you don't understand, really, the things will become really clumsy and they will become really very difficult. And then they say, right, that biochemistry is difficult. But biochemistry is really very easy if you understand the big picture. What is that big picture? The first page. Like all that food is oxidized. All that food is oxidized to acetyl-CoA. And that acetyl-CoA enters into the Krebs cycle, and from the Krebs cycle, reduced substrate go to the electron transport chain to produce energy. Well-fed state, insulin is the hormone, and that dumps all the glucose and all the amino acids and chylomicrons into the cells, and inside the cells, they make different things. While in the fasting state, there are different hormones that are important to increase the glucose level in the blood, because glucose is essential for the survival of brain and red blood cells, right? And then again, in the fasting state, ketone bodies are synthesized. And here, note here, that is really very important point, diabetes. You see that in diabetes, we don't have insulin. In diabetes, we don't have insulin. So what's happening? Actually, the cells are depriving. Cells are in the fasting state. In the diabetes, there is no glucose entering into the cells. All that glucose is remaining in the blood. That is why the glucose level is very high. And cells are essentially behaving as if they are starving. So, in the diabetes, the cells are not getting energy from glucose, rather they are getting energy from the fatty acids. They are getting energy from the fatty acids. And when in the liver, the fatty acids are oxidized, they make ketone bodies. And then those ketone bodies cause ketoacidosis. They accumulate in the blood, and in the blood, ketone bodies, when accumulate, they cause acidosis. What are ketone bodies? There are three important ketone bodies, and number one is acetone. This is of least importance in terms of energy, right? But it is important in diagnosis because this gives a fruity odor to that diabetic patient or starving patient. And the second ketone body is acetoacetate, 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 right? This is the second ketone body. And the third ketone body is 3-hydroxy or beta hydroxy, 3-hydroxybutyrate, 3-hydroxybutyrate. These are the ketone bodies. And you see that 8, right? Whenever there is come 8, that is an acid, acid or 8. These are the same thing. Acetoacetate, 3-hydroxybutyrate. So these are the acids in fact. And when the acids are there in the blood, that is going to cause acidosis, right? I hope now you understand this big picture. And if you don't understand, watch this video again, again and again. First clear these concepts, and then this will become really very easy to understand all those pathways. And it will take kind of not more than 2 hours to study all those pathways, right? Thank you so much. I hope you enjoyed. Please give your feedback. Thank you. Bye bye.